Again, it's me again. So I'm just going to take you through a little tutorial about some key words. The tonicity of solutions. So we've got these three terms over here, which you'll come across quite often in this topic: hypertonic, isotonic, and hypotonic. So hypertonic tells us that there's a higher solute concentration in the solution rather than the living tissue that we were talking about. So, we recognise maybe this word here, isotonic, from some sports drinks here, which means equal amounts of solute in cell and solution. So the isotonic sports drink you're drinking, let's say here, would contain, let's make the solute particles a bit bigger. They're blue. I'm going to make them solute particles red this time. Okay, so solute particles red. Let's start with isotonic solution, like that. The rest is water, let's assume. Okay, and so we've got not a very high concentration of water, not a very low concentration of water. So if we have this potato tissue here, I'll just draw another one over the isotonic. Yeah, I've got a bit of potato, a bit of living tissue, and it's got roughly the same salt concentration in its cells. It wouldn't look like that because its potato tissue is split up into millions of different cells, all with its own osmotic potential, right? However, we'd take it sort of as an average that this bit of potato has very similar osmotic potential to, where's my pen gone, this solution. If I put it in here, it would be isotonic, right? Which means, let's put it in, see if I can just drag it in here. I don't think I can. I'll just draw it in. Here's my potato in here. Okay, with the same salt concentration as its surroundings, which then means what happens to water movement? We do get some water moving in, but we get exactly the same amount of water moving out across through those membranes in the cells in the tissue. Okay, so there's no change here in mass at all. And you can see with these red blood cells, when I put the red blood cells as an isotonic solution, they look normal. We've got water going in, we've got water going out, no problem. So if you wanted to transplant an organ, for example, across the city from one hospital to another, the solution that you would want to keep that organ in is an isotonic solution. That means the same osmotic potential as the tissue that you're transporting, or the organs, several tissues that you're transporting in that organ. All right, so have I still got my red pen? No. Here's my potato tissue. Same concentration, same potato. Here's my hypertonic solution. Now, hypertonic means higher solute in solution. So I've got a really concentrated solution here. This would be really bad for transporting organs in or storing. Maybe if you wanted to actually preserve tissue, vegetables, something like that, you might want this kind of solution. So if I put my potato in here, with its not very concentrated tissue, what we then get is water moving out, because we have now a low concentration of water inside. We've got a high concentration of water inside, or high water potential inside, low water potential outside. So we've got lots of water moving out of the potato and a little bit moving in, because there is always something moving in. There's just more moving out. So here we have a decrease mass of the potato. Okay, and that's really good if you want to preserve vegetables. You remove the water, increase the salt content. Actually, that's how a lot of fermentation takes place, preserving that vegetable, pickling, if you like. All right, so here's my last potato. This is a black one. That's not confusing. My hypotonic. Now, my hypotonic is a much lower solute solution. You get it now. Yep. And my potato. My potato is in the middle. Yeah. So I put it in there in my solution here. There's my potato with my salt in it. Okay. So where's the low water potential? Where's the high water potential? Well, I've got not so much water concentration in the potato. I've got a really high concentration of water outside the potato. So I've got high water potential here, low water potential here, and we know now the movement goes high to low, high to low, to low, and a little bit goes in. Oh, sorry, a little bit goes out. A little bit goes out. Sorry, that's wrong. Okay, so we have now a massive increase in mass. Alongside that increase in mass, if we are an animal cell, we do not have a cell wall surrounding it, our cells are more likely to burst. So we can do huge damage to animal cells if you're transplanting them across town, across cities, put them in a hypotonic solution, damage will occur, right? They will start to burst eventually because the cell membrane is not structurally capable of preventing that from happening. A plant cell, on the other hand, is the cell membrane, has this kind of like a cage over at the cell wall made of cellulose, like a sort of cage-like structure, which does let most stuff in and out, or almost all stuff in and out. It's the membrane that controls what goes in and out. Okay, there we go. Hypertonic, isotonic, hypotonic, explained.